Welcome, curious minds, to another deep dive. Have you ever paused to truly appreciate the incredible array of plant life around us? From the tiniest specks of green in a pond to, well, the colossal trees forming entire forests, plants are not just wallpaper for our world. They are absolutely fundamental. And understanding this vast kingdom, especially its intricate classification and unique characteristics, is not merely for the scientifically curious. For many of you preparing for crucial examinations like NEET or your board exams, mastering this topic is absolutely foundational for academic success. Today, we're going to embark on a meticulous exploration of the plant kingdom, dissecting the classification and characteristics of its various groups, aiming to provide you with a clear, structured understanding. Joining us to guide this essential learning journey is our expert.
Thank you. Yes, I'm ready to delve into this detailed exploration of the plant kingdom, focusing specifically on Chapter 3 from your syllabus. Our objective is to provide you with a thorough, exam-oriented understanding, much like, a focused lecture you'd experience in a coaching institute. We'll aim to ensure every concept is clear, every distinction highlighted, and that you feel, properly equipped to confidently approach any question on this topic in your upcoming exams.
All right, let's begin by setting the stage. Many of us recall R.H. Whitaker's five-kingdom classification from earlier studies, but our scientific understanding of what precisely constitutes the plant kingdom has certainly evolved since then, hasn't it? Could you perhaps elaborate on how this modern perception of plantae differs from earlier views and what that means for our current syllabus?
That's a perceptive question, absolutely. And understanding this evolution is indeed a crucial starting point for everyone. Let's briefly revisit Whitaker's five-kingdom classification, proposed back in 1969. This system categorized living organisms into five broad kingdoms: Monera, Protista, Fungi, Animalia, and Plantae. That's the framework you likely learned first. While this provided a robust framework, our scientific knowledge, which is continuously advanced by new research and, better tools, has refined the boundaries of what we specifically include under Kingdom Plantae for your curriculum. Historically, it's important to note that groups like fungi and certain members of Monera and Protista that happen to possess cell walls were often lumped into Plantae. However, a key point for your understanding, especially for competitive exams, is that these groups are now explicitly excluded from Plantae. Take, for instance, cyanobacteria. These were once commonly, though, incorrectly referred to as blue-green algae. But in the modern accepted classification of Plantae, cyanobacteria are no longer considered true algae in the context of this chapter, nor are they part of the plant kingdom proper. So for our deep dive today, and critically for your NEET and board exam syllabus, when we refer to Plantae, we will be specifically describing five major groups: algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. This precise definition is really important to avoid confusion.
That's a really important clarification, definitely, especially concerning cyanobacteria. That's a common confusion point. Now, moving beyond the broad kingdom definitions, even within the classification of plants themselves, the systems have seen significant evolution.
Right.
Can you walk us through the different approaches to plant classification, maybe from the earliest to the most current, explaining sort of why each system arose?
Absolutely. Tracing the evolution of these classification systems is fundamental to appreciating the robustness of our current understanding. Let's trace this progression step by step. The earliest systems were what we call artificial systems. These were quite rudimentary, relying solely on gross superficial morphological characters. Think about it. They use easily observable features like the habit of the plant, whether it was a herb, shrub, or tree, its color, the number and shape of its leaves, things like that. A classic example is the system proposed by Carolus Linnaeus, which primarily focused on vegetative characters, or quite specifically, the structure of the androecium, which is the male reproductive part of the flower. Now, these artificial systems had significant limitations, and understanding these drawbacks is actually crucial for your exams. Because they relied on only a few easily observable characteristics, they often ended up separating closely related species. Imagine two species that are genetically very similar, but perhaps one grows in a sunny, dry area and develops smaller leaves, while the other is in a shaded, moist spot and has larger leaves. An artificial system, by giving too much weight to such, variable features, might mistakenly place them in different groups. This is why these systems ultimately proved inadequate.
I see. Could you clarify for our students why giving equal weight to vegetative and sexual characteristics in these artificial systems proved to be such a significant drawback? It seems like a point that could easily confuse someone if they don't grasp the underlying reason.
That's an excellent follow-up question, and it addresses a core conceptual challenge. Yes, The major drawback of artificial systems giving equal weight to both vegetative and sexual characteristics stems from a fundamental biological reality. Vegetative characters are often much more easily affected by environmental factors, whereas sexual characteristics are generally more stable and genetically conserved. Let me elaborate a bit. Vegetative characters include features like the size and shape of leaves, the overall growth habit, or even the color of the plant's non-reproductive parts. These traits can vary significantly depending on external conditions, things like light availability, nutrient levels in the soil, water scarcity, or temperature. For instance, a plant grown in a shaded, nutrient-rich environment might develop larger, lusher leaves than the exact same species grown in harsh, sunny, nutrient-poor conditions. The environment directly influences these traits, making them quite well plastic. On the other hand, sexual characteristics, particularly the intricate structure of reproductive organs and the precise process of reproduction, are generally much more stable. They are deeply ingrained in the plant's genetic makeup and are often highly conserved across closely related species. This stability makes them, better indicators of true evolutionary relationships. By treating an environmentally variable vegetative trait with the same importance as a stable sexual trait, artificial systems could inadvertently group unrelated plants together, or conversely, separate truly related ones based purely on superficial variable features. This led to an unnatural classification that did not accurately reflect the true biological affinities or evolutionary history of the organisms, making it unreliable for predicting broader relationships or for deeper scientific study. This fundamental flaw is why we eventually moved beyond them.
That's a very clear explanation, thank you. So if artificial systems had such inherent limitations, what advancements came next to overcome these issues? What was the next step?
Right. So to overcome these limitations, natural classification systems were developed. These systems marked a really significant leap forward in taxonomy. Instead of just superficial characters, they were based on natural affinities among organisms. This meant considering a much broader spectrum of features, including not only external morphology, but also a wealth of internal features. For example, they looked at the ultrastructure of cells, those minute details visible only with powerful microscopes. They delved into the anatomy of the plant, so internal tissue organization, its embryology, which is the development of the embryo, and even its phytochemistry, the chemical constituents found within the plant. This more holistic approach allowed for a much more accurate representation of relationships. A well-known example of a natural classification for flowering plants was provided by George Bentham and Joseph Dalton Hooker. It's a system still referred to even today for certain purposes, actually. Following the natural systems, we now primarily use phylogenetic classification systems. These are currently the most acceptable and widely used systems in biology. Their fundamental basis is the evolutionary relationships between organisms. The core assumption here is a powerful one. Organisms belonging to the same taxa or groups share a common ancestor. This system aims to reflect the evolutionary history, the phylogeny of plants. What's truly fascinating is how information from many diverse sources is integrated to resolve classification difficulties, especially when clear fossil evidence, which would directly show evolutionary links, is, lacking. This brings us to the modern tools in taxonomy, which are incredibly powerful for creating these comprehensive phylogenetic classifications. First, we have numerical taxonomy. This method is now easily carried out using computers, which makes it very efficient. It operates by considering all observable characteristics of an organism. Each character is assigned a number and a code, and then the data is processed mathematically, statistically. The beauty of this system is that every single character, whether it's the number of petals, the specific type of leaf venation, or the presence of a certain chemical compound, is given equal importance. This objective approach allows taxonomists to simultaneously consider hundreds, even thousands, of characters, leading to a much more objective and data-rich classification. Second is cytotaxonomy. As the name suggests, this approach is based on cytological information. This includes details like the precise chromosome number within the cells, their intricate structure, how they're shaped, if there are any unique banding patterns, and their behavior during cell division, particularly meiosis. Deviations or similarities in these chromosomal characteristics can provide strong clues about evolutionary relationships, especially among closely related species. And finally, we have chemotaxonomy. This method leverages the chemical constituents of the plant. Different plant groups produce unique chemical compounds, often known as secondary metabolites. By analyzing these biochemical fingerprints, things like specific alkaloids, terpenes, flavonoids, or even unique proteins and amino acids, taxonomists can resolve confusions and establish clearer relationships. For instance, the presence or absence of a particular chemical might be diagnostic for a specific plant family. You see, these modern approaches, when combined, provide a far more comprehensive and accurate classification system than anything that came before. This multifaceted approach, integrating morphological, anatomical, embryological, phytochemical, cytological, and evolutionary data, is absolutely essential knowledge for your competitive exams, as questions often delve into the underlying principles and applications of these classification methods.
Right. For NEET aspirants, understanding these different approaches is certainly a key area for questions. Could you maybe briefly summarize the core principle of each modern tool like numerical taxonomy and chemotaxonomy, and perhaps where they apply best? It would be great to distill this down for quick revision.
That's an excellent idea, yes. A good way to consolidate for exam preparation. Let's distill this down for quick recall. Okay, for numerical taxonomy, the core principle is quantification and equal weighting of all observable characteristics. You assign numbers and codes to every single feature, whether it's a macroscopic trait like the number of leaves or a microscopic one like pollen grain morphology. This data is then fed into computers. Its best application is when you have a vast amount of data and you want an objective, unbiased classification that considers hundreds of characters simultaneously without subjective human judgment influencing which characters are more important. It helps in drawing overall similarities efficiently. For cytotaxonomy, here, the core principle is using internal cellular details, specifically related to the chromosomes. This means looking at the chromosome number, diploid, polyploid, their precise structure, like looking for inversions or translocations, and their behavior during cell division, like meiosis. It applies best when trying to establish relationships between groups that might look morphologically similar, but have underlying genetic differences, or to understand evolutionary divergences at a chromosomal level. It offers insights into genetic affinities that external features might not reveal. And for chemotaxonomy, the core principle here is using the unique chemical fingerprint of a plant. Different plants produce specific chemical compounds, often secondary metabolites, that are unique to their lineage. By identifying and comparing these chemical constituents, things like specific alkaloids, flavonoids, or unique amino acids, taxonomists can determine relationships. This applies best when morphological or anatomical data might be ambiguous, or when trying to differentiate between, say, cryptic species that look very similar but have distinct biochemical pathways. It provides a deeper biochemical insight into relationships. So, in essence, numerical taxonomy gives a broad, objective overview. Cytotaxonomy provides genetic insights, and chemotaxonomy gives a biochemical perspective. Together, they create a much clearer picture, making them invaluable tools.
That's a fantastic and succinct summary. Very clear for exam purposes. Excellent. Now let's dive into the first major group within Plantae that we'll be discussing, algae. These are such diverse and fundamental life forms, aren't they? Especially in aquatic ecosystems. What are their general characteristics and just how varied are they in form and size?
All right, let's transition to algae. Yes, they are truly foundational to many ecosystems and represent some of the earliest photosynthetic life forms we know. When we talk about algae, we're referring to organisms that are, first and foremost, chlorophyll-bearing, meaning they perform photosynthesis just like higher plants. They're typically simple in structure, exhibiting what we call a thalloid body plan. This means their body is undifferentiated. You cannot distinguish true roots, stems, or leaves, unlike the more complex plants we'll discuss later. It's just a thallus. They are autotrophic, producing their own food using sunlight, and they are largely aquatic, found abundantly in both freshwater bodies like ponds and lakes and vast marine environments like oceans. But their habitats aren't strictly limited to water, actually. You can also find them in a variety of other moist places, on damp stones, in moist soils, and even on wood. Interestingly, some algae form fascinating symbiotic associations. A classic example is their association with fungi, forming what we know as lichens, which are often pioneer colonizers on rocks. Another intriguing instance is their association with animals, like the green algae found growing in the fur of a sloth bear, giving it a greenish tinge that provides camouflage. Quite interesting. In terms of their form and size, algae show an astonishing range, a point often tested for examples. They can be incredibly tiny, existing as unicellular forms like Chlamydomonas, which you'd need a microscope to see properly. Then you have colonial forms, where many cells live together in a coordinated colony, a classic example being Volvox, which often forms beautiful spherical colonies. Moving up in complexity, some are filamentous forms, creating long thread-like structures, such as Eleutheryx and Spirogyra. And then at the other end of the spectrum, we have the truly massive marine forms like kelps, which are a type of brown algae. These kelps can be incredibly large, forming vast underwater forests and reaching an astounding height of up to 100 meters. Imagine a plant taller than a 30-story building. This truly showcases the immense and diverse scale within this group.
That range is incredible, from microscopic to 100-meter giants, really quite something. Given this diversity, how do these various forms of algae reproduce? Could you break down their vegetative, asexual, and sexual reproduction methods, especially highlighting the sexual distinctions, which, as you know, are often a point of confusion for students in exams?
Yes, that's a very important area, as understanding the reproductive strategies is key for exam success. Algae employ all three major methods of reproduction: vegetative, asexual, and sexual. Let's take this step by step. First, vegetative reproduction. This is the simplest and perhaps most common method, occurring primarily by fragmentation. What happens is that the phallus, the plant body, simply breaks into several pieces, and each fragment has the remarkable capacity to develop into a complete new thallus. It's a very efficient way for them to rapidly increase their numbers, especially in favorable conditions. Second, asexual reproduction. This typically involves the production of different types of spores. The most common and frequently encountered type of asexual spore in algae is the zoospore. Zoospores are distinct because they are flagellated, meaning they possess flagella, these whip-like structures that enable them to be modal or capable of self-movement in water. Once these zoospores are released, they can swim around, find a suitable substrate, and then germinate directly to give rise to new plants. So you can imagine them swimming through the water, finding a good spot, and starting a new algal growth. Third, and perhaps the most complex for distinctions, is sexual reproduction. This method involves the fusion of two gametes. The classification here is crucial for understanding evolutionary progression and is frequently tested, so pay close attention to the gamete characteristics. 1. Isogamous reproduction. In this type, the two fusing gametes are similar in size. Now here's an important nuance. They can either be flagellated and similar in size, as seen in Eulothrix, meaning both male and female gametes look identical and can swim, or they can be non-flagellated, non-modal, but still similar in size, which is characteristic of Spirogyra. The key distinguishing feature here for both types is that the fusing gametes are similar in size. Iso means same. 2. Anisogamous reproduction. This is where the gametes start to differ. Here you have the fusion of two gametes that are dissimilar in size. One gamete is typically noticeably larger than the other. An example of this can be found in certain species of Euderina. They might still both be modal, but their sizes are clearly distinct. Aniso means unequal. 3. Oogamous reproduction. This represents the most advanced type of sexual reproduction observed in algae, and it is a significant evolutionary step. In oogogamy, there is a very clear and pronounced distinction between the male and female gametes. You have a large non-modal static female gamete, which is often referred to as an egg or ovum. This large sessile female gamete is fertilized by a smaller modal male gamete. Classic examples of oogamous reproduction are observed in Volvox and Fucus.
Okay, this classification of sexual reproduction is indeed very important for students. It comes up a lot. Can you maybe give a quick memorable analogy or perhaps a trick for students to differentiate between isogamous, antisogamous, and allogamous types in their minds, really focusing on those gamete characteristics? This often trips up students in MCQs.
That's a great idea, definitely. Let's create some simple mental images to help recall these distinctions easily for your exams. Think of it like a dance party for gametes, focusing on the partners involved. For isogamous, imagine identical twins dancing together. They're exactly the same size and look, and crucially, they either both can move freely, if flagellated like Eulothrix, or neither can move if non-flagellated like Spirogyra. The key point is, they are identical in appearance and size. Remember, iso means same, so isogamous means same gametes in terms of size and, if motile, motility, for anisogamous. Now, imagine a dance where the partners are clearly different sizes. Maybe one is a bit taller or wider than the other, but both are still actively moving and participating. They're clearly distinct in size, but often still share the characteristic of being motile. Remember, aniso means unequal, so anisogamous means unequal gametes in terms of size. And for oogamous, this is like a very traditional or formal waltz, perhaps. You have one large, elegant partner who remains stationary. This is your large, non-motile female gamete, the egg. And then you have a smaller, nimble, very active partner who comes to where this is your small, modal male gamete. The roles are very distinct. One is clearly static and large, while the other is mobile and small. The prefix oo often relates to egg, and eggs are typically large and non-modal, so you can remember. Identical twins for isogamous, unequal-sized dancers for anisogamous, and static large partner and nimble small partner for oogamous. That visual cue should help you quickly recall the key differences in gamete size and motility, which are definitely frequently tested.
Those analogies are very effective. Yes, I think that will surely help students remember the distinctions much better. Now, beyond their reproduction, algae also have significant economic importance, which, as you mentioned, is often a high-yield topic for exams. What are some of the key ways algae are useful to humans and ecosystems?
Indeed, their economic and ecological importance cannot be overstated, and this is a common area for questions. Absolutely. Algae contribute to our planet and to human welfare in a variety of crucial ways. First and foremost, algae are incredibly vital as primary producers. They are responsible for carrying out at least half of the total carbon dioxide fixation on Earth through photosynthesis. Think about that, half. This massive photosynthetic activity means they absorb tremendous amounts of CO2 from the atmosphere and convert it into organic compounds, essentially forming the base of the global food chain. As a direct result of this photosynthesis, they significantly increase the level of dissolved oxygen in their immediate aquatic environment, which is indispensable for other aquatic life, like fish and invertebrates. Essentially, they form the fundamental basis of the food cycles for all aquatic animals, serving as the primary producers of energy-rich compounds that sustain entire food webs. Beyond their ecological role, many species of marine algae are directly consumed as food sources, especially in East Asian cultures, but elsewhere too. It's estimated that about 70 species of marine algae are used as food globally. Notable examples include porphyra, which is the source of nori used in sushi wraps, laminaria, and sargassum. Furthermore, chlorella, a unicellular alga, is remarkably rich in proteins and is so nutritious that it's even used as a food supplement, famously by space travelers during long missions to ensure a compact, nutrient-dense food source. Then there are the commercial hydrocolloids. These are essentially water-holding substances produced by certain marine brown and red algae, and they have significant commercial applications in various industries. Algin is obtained specifically from brown algae. It's widely used in industries as a thickener, emulsifier, and stabilizer in products like ice cream, giving it that smooth texture, cosmetics, and textiles. Carrageen is another important hydrocolloid derived from red algae. It's extensively used in the food industry as a gelling, thickening, and stabilizing agent, particularly in dairy products like milk and ice cream. And finally, agar, which is one of the most well-known commercial products, is obtained from specific red algae, primarily Gelidium and Gracilaria. Agar is indispensable in microbiology labs as a solidifying agent to grow microbes in petri dishes. It provides a firm medium for bacteria and fungi. It's also a common ingredient in the preparation of ice creams and jellies, giving them their characteristic texture. So when you consider their role in the global carbon cycle, their direct use as food, and their versatile industrial applications, the economic importance of algae is truly immense and definitely a very important topic for your exams.
It's fascinating how these seemingly simple organisms play such a massive role, both ecologically and economically, really underlines their importance. Now, you mentioned three main classes of algae: Chlorophyceae, Phaeophyceae, and Rhodophyceae. For our students, getting the distinguishing features of each class down is crucial for MCQs and short answers. Could you delve into each of these classes, highlighting their common names, pigments, food storage, cell wall, and reproductive characteristics, almost as if we're going through a detailed comparative table, perhaps referencing Table 3.1 from the textbook?
Absolutely. Let's break down the three main classes of algae in detail, as differentiation between them is a frequent exam question. I encourage you to visualize a comparative table, much like Table 3.1 in your textbook, as we go through these points. It's a great study aid. First, 3.1.1 Chlorophyceae, commonly known as green algae, common name. Green algae, pretty straightforward. Plant body. Their body can be quite varied. You'll find unicellular forms like Chlamydomonas, colonial forms such as Volvox, forming those spherical colonies, and filamentous forms including Elothrix and Spirogyra, which is famous for its spiral chloroplasts. Pigments. Their characteristic grass-green color comes from the dominance of chlorophyll A and chlorophyll B pigments. These pigments are specifically localized within definite chloroplasts inside the cells, allowing efficient photosynthesis. Chloroplast shapes. This is a distinctive feature, actually, and a common MCQ detail. Chloroplasts can come in various shapes depending on the species. They might be discoid, plate-like, reticulate, like a net, cup-shaped, as in Chlamydomonas, spiral, as in Spirogyra, or even ribbon-shaped. This species-specific variation is an important detail to note. Storage bodies. Most members possess one or more specialized storage bodies called pyrenoids, which are located within their chloroplasts. Pyrenoids primarily contain protein, but they also store starch around them. In some green algae, food might also be stored in the form of oil droplets. Cell wall. Green algae typically have a rigid cell wall, which is composed of two layers, an inner layer made of cellulose and an outer layer made of pectose. Reproduction. Vegetative reproduction usually occurs by fragmentation, simple breaking apart. Asexual reproduction is primarily by flagellated zoospores, which are produced within specialized structures called zoosporangia. Sexual reproduction shows considerable variation in the type and formation of sex cells, encompassing all three types we discussed: isogamous, anisogamous, or oogamous. Examples: some commonly found green algae include Chlamydomonas, Volvox, Eulothrix, Spirogyra, and Chara. Knowing these examples is key. Next, let's move to 3.1.2 Phyophyceae, known as brown algae, common name. Brown algae. Habitat. They are found primarily in marine habitats, dominating many coastal areas and often forming those extensive kelp forests. Size and form. They display remarkable variation in size and form, ranging from simple branched filamentous forms like ectocarpus to profusely branched, massive forms represented by the kelps, which, as we mentioned, can reach that impressive height of 100 meters. Pigments. Brown algae possess chlorophyll A and chlorophyll C, along with carotenoids and xanthophylls. Their characteristic color, which ranges from olive green to various shades of brown, is due to the varying amount of a specific xanthophyll pigment called fucoxanthin present in them. This fucoxanthin allows them to absorb a wider spectrum of light, especially in deeper waters. Stored food. Food is stored as complex carbohydrates, which specifically take the form of laminarin or mannitol. These are unique storage polysaccharides different from starch. Cell wall. Their vegetative cells have a cellulosic wall, which is typically covered on the outside by a gelatinous coating of algin, the hydrocolloid we discussed earlier, giving them that characteristic slimy feel. Protoplast. The protoplast within their cells contains plastids, usually a central located vacuole and a nucleus. Plant body structure. Their plant body is usually well differentiated compared to many green algae. It is typically attached to the substratum by a structure called a holdfast, which anchors it firmly. It then has a stalk-like structure known as the stipe, and a leaf-like photosynthetic organ called the frond. This holdfast, stipe, and frond differentiation is important for diagram-based questions. Reproduction. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae involves biflagellate zoospores. These zoospores are distinctively pear-shaped, pariform, and possess two unequal flagella that are attached laterally on the side. This pear-shaped and unequal lateral flagella are a key distinguishing feature often asked in MCQs. Remember that. Pear-shaped, two unequal lateral flagella. Asexual reproduction in most brown algae involves biflagellate zoospores. These zoospores are distinctively pear-shaped and possess two unequal flagella that are attached laterally on the side. This pear-shaped and unequal lateral flagella are a key distinguishing feature often asked in MCQs. Remember that. Sexual reproduction can be isogamous, anisogamous, or oogamous. The union of gametes may occur directly in the water or within the Oogonium and Oogamous species. The gametes themselves are also pyriform, or pear-shaped, and bear two laterally attached flagella, similar to their zoospores. Examples. Common forms include Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. Again, knowing these is important. Finally, let's cover 3.1.3 Rhodophyceae, or red algae, common name, red algae, pigment. Their striking red color is due to the predominance of a specific red pigment called R-phycoerythrin in their body. This pigment is crucial because it allows them to absorb blue-green light, which penetrates deeper into the ocean than other light wavelengths. Habitat. The majority of red algae are marine, and they show greater concentrations in warmer ocean areas. What's particularly interesting is their adaptability. They're found in both well-lighted regions close to the water's surface and at great depths in oceans where relatively little light can penetrate. This ability to thrive in low-light conditions is a direct result of their unique red pigments, which are highly efficient at absorbing those specific wavelengths, a remarkable adaptation. Thallus. The red thalli of most red algae are multicellular, and some species exhibit surprisingly complex body organization, unlike the simpler thalli of many green algae. Stored food. Their food is stored in the form of Floridian starch, which is structurally very similar to amylopectin and glycogen, common storage carbohydrates found in animals and fungi. This biochemical similarity is a unique detail often asked in competitive exams. Remember Floridian starch for red algae. Reproduction. Vegetative reproduction typically occurs by fragmentation. Asexually, they reproduce by non-motile spores. This is a key difference from green and brown algae. No flagella here. Sexually, they reproduce by non-motile gametes as well. Sexual reproduction in red algae is exclusively oogamous and is often accompanied by complex post-fertilization developments, making their life cycle quite intricate. So non-motile spores and non-motile gametes are characteristic of red algae. Common members include Polysiphonia, Porphyra, also a food source, remember nori, Gracilaria, and Ptillidium. Remember, Gracilaria and Ptillidium are the primary sources of agar. So by paying close attention to these specific features, the dominant pigment and its ecological role, the type of stored food, the flagellation or lack thereof of spores and gametes, and their typical habitat, you can clearly differentiate between these three classes for your exams. That comparative table in your textbook is really an excellent, quick revision tool for these points.
That detailed breakdown is incredibly helpful, especially emphasizing those unique features like chloroplast shapes, specific flagella types, and even the nuances of pigment function for adaptation in red algae. That's great. Now, moving on from the algae, let's talk about the bryophytes, often called the amphibians of the plant kingdom. Why are they given this unique designation, and what are their defining characteristics?
Right. The term amphibians of the plant kingdom for bryophytes is a key concept, definitely frequently asked in exams, because it highlights a fundamental aspect of their biology and their evolutionary position. Bryophytes include various mosses and liverworts, which you commonly find growing in moist, shaded areas, especially in hilly regions. Think damp walls, shady banks. They are called the amphibians of the plant kingdom, precisely because, while they can live on soil and show adaptations to terrestrial life in terms of their body structure, they're absolutely dependent on water for sexual reproduction. Specifically, water is required for the transfer of their male gametes, called antherozoids, to the female sex organs. Without a film of water for these antherozoids to swim through, fertilization simply cannot occur. This dependency on water for reproduction is what fundamentally limits their habitat to damp, humid, and shaded localities, despite their terrestrial presence. They need that moisture. Despite this water dependency, bryophytes play an important ecological role that is often examined in applying questions. They are significant as pioneers in succession. Along with lichens, they are often among the very first organisms to colonize bare rocks in newly formed soil. They initiate ecological change in such barren environments. Through their growth and subsequent decomposition, they contribute to decomposing rocks, helping to create and enrich the substrate, making it more suitable for the growth of higher plants later on. So, soil formation. Mosses, in particular, form dense mats on the soil surface. This dense growth effectively reduces the impact of falling raindrops and thus significantly helps to prevent soil erosion, especially on slopes. That's an important environmental function. A notable example is sphagnum, a type of moss which provides peat. Peat has been used as a fuel for centuries, particularly in regions where wood is scarce. More importantly, for commercial purposes, sphagnum has an astonishingly high water-holding capacity. It can absorb many times its own weight in water. This property makes it an excellent packing material for the transshipment of living plants, seedlings, and other biological materials, keeping them moist and viable over long distances. Very useful. Furthermore, some mosses also provide food for herbaceous mammals, birds, and other animals in certain ecosystems.
So their amphibian nature truly revolves around that reproductive constraint, the need for water. Okay. How does their plant body differentiate from algae, and what's unique about their life cycle? Especially the dominant phase, which is a key distinction from other plant groups, right?
Yes, exactly. Let's break down their plant body and life cycle, as these are critical areas for distinguishing bryophytes. The plant body of bryophytes shows a more significant differentiation compared to that of algae. While still relatively simple, it's not just an aquatic thallus. Instead, it is typically phallus-like, but can be either prostrate, lying flat on the ground, like many liverworts, or erect, like most mosses. This body is attached to the substratum, the soil or rock-based structures called rhizoids. These rhizoids can be unicellular or multicellular. It's crucial to remember they are not true roots. They primarily function in anchorage and some absorption of water and minerals from the surface, but they lack the complex vascular tissues, xylem and phloem, found in true roots. In fact, bryophytes generally lack true roots, stem, or leaves, although they may possess root-like, leaf-like, or stem-like structures. These are evolutionary advancements over algae, but not yet fully differentiated organs like you see in higher plants. Now, for their life cycle, this is a key differentiator that is frequently tested. The main plant body of a bryophyte is haploid, meaning its cells contain a single set of chromosomes, N. This dominant, free-living, and photosynthetic stage is called the gametophyte, and its primary role is to produce gametes. Remember, dominant gametophyte. Let's look at the sex organs, which are multicellular in bryophytes. The male sex organ is called an antheridium, plural antheridia. The antheridium produces biflagellate antherozoids, which are the motile male gametes, equipped with two flagellas enabling them to swim in water. The female sex organ is called an archegonium, plural archegonia. It is distinctively flask-shaped and produces a single egg, which is the non-motile female gamete, located at its base within the flask. Here's how fertilization happens. The antherozoids are released into the film of water present in their habitat. They then swim towards and come into contact with the archegonium. An antherozoid then fuses with the egg inside the archegonium to produce a zygote, which is now diploid, 2MA. Now, this is where the life cycle becomes truly distinctive and a point of frequent examination. The zygotes in bryophytes do not undergo reduction division meiosis immediately. This is different from some algae. Instead, they undergo mitotic divisions to develop into a multicellular body called a sporophyte, still 2N. The sporophyte is typically differentiated into three parts: a foot, which is embedded in the gamophyte tissue, a seta, a stalk, and a capsule, the spore-producing part at the top. A critical concept here, and a frequently asked question in competitive exams, is that the sporophyte is not free-living. It remains permanently attached to the photosynthetic gamophyte and is completely dependent on it for nourishment, essentially functioning as a parasite on the gamophyte. It cannot survive on its own. Within the sporophyte, specifically in the capsule, some cells called spore mother cells undergo reduction division, or meiosis, to produce haploid spores N. These spores are then dispersed, often by wind or specialized mechanisms. Finally, these haploid spores germinate under suitable moist conditions to produce a new gamophyte, and thus completing the life cycle. So the dominant phase in bryophytes is the haploid gamophyte, which bears the dependent diploid sporophyte.
Right, the dependence of the sporophyte on the gametophyte in bryophytes is a critical concept, as you just highlighted. Could you maybe reiterate why this is so significant in their life cycle for our students? What does it really imply about their evolutionary standing or perhaps their ecological limitations?
Yes, this point is absolutely crucial for understanding bryophytes, and is a common area for conceptual questions. The fact that the sporophyte in bryophytes is not free-living and remains attached to and entirely dependent on the photosynthetic gamophyte for its nourishment is profoundly significant for several reasons. First, from an evolutionary perspective, it tells us that bryophytes represent an early, transitional stage in the evolution of land plants. While they have indeed evolved a more differentiated body plan than algae and are adapted to live on land, they haven't achieved full independence of the sporophyte stage from the gametophyte stage. Remember, the gametophyte is the dominant photosynthetic and free-living phase. This dependency of the sporophyte signifies a less advanced state compared to higher plants, like Pteridophytes, Gymnosperms, and Angiosperms, where the sporophyte becomes the dominant, independent, and photosynthetic plant body. It indicates that the major evolutionary trend in plants, the shift from a dominant gametophyte to a dominant sporophyte, is still, well, incomplete in bryophytes. It's a stepping stone. Second, in terms of ecological limitations, this dependency directly reinforces their amphibian nature and explains their restricted distribution. Since the sporophyte relies entirely on the gametophyte, and the gametophyte's sexual reproduction requires external water, the entire life cycle remains inextricably tied to moist environments. The sporophyte cannot simply germinate and grow independently anywhere. This limits the geographical distribution of bryophytes primarily to damp, humid, and shaded localities, as the success of their entire life cycle hinges on that critical connection and the continuous availability of a film of water for fertilization. Without that photosynthetic gametophyte supporting the sporophyte, the production of spores, and thus the continuation of the species, would not be possible. It's a significant bottleneck in their complete adaptation to truly dry terrestrial environments.
That clarifies the implications perfectly. Thank you. Now, let's look at the two main divisions within bryophytes, liverworts and mosses. What are their distinct features, particularly in terms of their body structure and maybe asexual reproduction? These distinctions are often asked in comparative questions, aren't they?
Certainly. Bryophytes are broadly divided into liverworts and mosses, each with unique characteristics that are important for differentiation in your exams. Let's begin with 3.2.1 liverworts. Habitat. Liverworts typically grow in very moist, shady habitats. You'll find them in places like the banks of streams, on marshy ground and damp soil, on the bark of trees, and deep within forests where light is scarce and humidity is high. Very specific habitats. Plant body. The plant body of a liverwort is generally thalloid, meaning it's a simple, undifferentiated structure. A classic example is Marchantia. The thallus is distinctly dorsiventral, which means it has a flattened upper dorsal surface and a lower ventral surface, and it lies closely pressed flat, to the substrate. Some liverworts are called leafy members, meaning they have tiny, delicate leaf-like appendages arranged in two rows on stem-like structures, although these are not true leaves with vascular tissue. Asexual reproduction. This is a key distinguishing feature and a common MCQ point. Asexual reproduction in liverworts can occur by fragmentation of their thalli. But more famously, it occurs by the formation of specialized structures called gemmo, singular.gemmo. Gemma are remarkable. They are green, multicellular, asexual buds. These gemma develop within small, cup-shaped receptacles found on the dorsal surface of the thalli, aptly named gemma cups. When these gemma detach from the parent body, perhaps by a splash of rain, they are capable of germinating to form new, genetically identical individuals. Look out for gemma cups in diagrams. Sexual reproduction. During sexual reproduction, the male and female sex organs can be produced either on the same thallus, monoecious, or on different thalli, dioecious. The sporophyte that develops from the zygote is relatively simple compared to mosses, usually differentiated into a foot, seta, and capsule. After meiosis occurs within the capsule, haploid spores are produced. These spores are then released and germinate to form new, free-living gamophytes. Now let's move to 3.2.2 mosses, predominant stage. The predominant stage in the life cycle of a moss is the gamophyte, just like in liverworts. However, the gamophyte of a moss is generally more elaborate and consists of two distinct stages. 1. The first stage is the protonema stage. This develops directly from a germinating spore. It's a creeping, green, branched, and frequently filamentous stage, somewhat resembling an algal filament in appearance. This is the juvenile stage. 2. The second stage is the leafy stage. This develops from the secondary protonema as a lateral bud. This stage is what you typically recognize as a moss plant. It consists of upright, slender axes bearing spirally arranged leaves. These leafy shoots are attached to the soil by multicellular and branched rhizoids, which are generally more complex than the unicellular rhizoids of many liverworts. Crucially, this leafy stage is where the sex organs are born, usually at the apex. Vegetative reproduction. In mosses, vegetative reproduction primarily occurs by fragmentation of the secondary protonema and by budding in the secondary protonema. The secondary protonema can easily fragment, and new buds can sprout from it to form new plants. Sexual reproduction. The sex organs, Antheridia, male, and Archegonia, female, are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte, which is significantly more elaborate than that found in liverworts. It also consists of a foot, seta, and capsule. The capsule contains the spores, which are formed after meiosis. Mosses are also known for having an elaborate mechanism of spore dispersal, often involving a structure called a peristome, which is a ring of teeth-like structures around the capsule mouth that respond to humidity changes, helping release spores gradually and effectively over a wider area. That's a detail often observed under a microscope or in diagrams. Examples. Common examples of mosses include Funaria, often called the common moss, Polytrichum, and Sphagnum, the peat moss. So, while both are bryophytes sharing the gametophyte-dominant, water-dependent reproduction, mosses generally have a more complex gametophyte with those distinct protonema and leafy stages, and their sporophyte is typically more elaborate and efficient in spore dispersal than that of liverworts. This differentiation is a classic comparison point for your exams.
That clear distinction between liverworts and mosses, particularly that protonema stage in mosses, is excellent for memorization and understanding. Definitely. Now, we're talking about further land adaptations, right? And that brings us to pteridophytes, often called the first terrestrial vascular plants. What makes them such an evolutionary leap? And how does their dominant phase and plant body structure significantly differ from bryophytes?
Yes, exactly. We're now moving to a very significant evolutionary step in the plant kingdom with pteridophytes, which include familiar forms like horsetails and ferns. Their designation as the first terrestrial vascular plants is arguably their most important defining feature and a frequently asked question in competitive exams. You really need to know this. This is a truly critical point. Pteridophytes are the first terrestrial plants to possess vascular tissues specifically, xylem and phloem. Recall that xylem is responsible for efficient water and mineral transport from roots up to all parts of the plant, and phloem for the transport of food, sugars produced during photosynthesis, throughout the plant body. The development of these specialized conducting tissues was a monumental evolutionary leap. It allowed plants to grow much larger in size and provided a much more efficient system for transporting water and nutrients, enabling much better adaptation to a true land-based existence, away from constant immersion in water. This feature allowed them to explore and colonize more diverse terrestrial habitats compared to bryophytes. You'll study these tissues in much more detail in your plant anatomy chapter, of course. Pteridophytes are not just evolutionarily significant. They are also used for various practical purposes. They have medicinal uses in traditional systems, act as excellent soil binders preventing erosion on slopes, and are frequently grown as beautiful ornamentals in gardens due to their graceful fronds, like ferns. While they represent a major terrestrial adaptation, they're still typically found in cool, damp, and shady places. though some species have adapted to flourish in sandy soil conditions, showcasing some resilience. Now, let's contrast their dominant phase and plant body with bryophytes. You may recall that in bryophytes, the dominant and free-living phase in the life cycle is the haploid gamophytic plant body. However, in pteridophytes, a major evolutionary shift occurs. The main plant body is a sporophyte, diploid two-butted, and it is fully differentiated into true roots, true stems, and true leaves. This is a fundamental distinction from bryophytes. These true organs, unlike the root-like or leaf-like structures of bryophytes, possess well-differentiated vascular tissues, allowing for their larger size, more complex structure, and greater independence from water for structural support and internal transport. Their leaves can vary in size. Some pteridophytes have small leaves, known as microphylls, as seen in Phylogenetic, also called spike moss. Others, like most ferns, have much larger leaves, referred to as macrophylls. This variation in leaf size, microphyll versus macrophyls, is also a point of distinction sometimes asked.
That shift to a dominant sporophyte with true vascular tissues is indeed a game changer, absolutely. How does their reproduction cycle work then, and what's the significance of this concept of homospore versus heterospore? Especially considering the evolution of the seed habit, that sounds like a critical concept for advanced studies and exams like NEET.
Yes, let's delve into their reproduction cycle and this crucial concept of homospore versus heterospore, as it's a fundamental evolutionary step often examined. The sporophytes and pteridophytes, being the dominant phase, bear sporangia, which are the structures that produce spores. These sporangia are subtended by, or borne on, specialized leaf-like appendages called sporophylls. In some pteridophytes, these sporophylls can form distinct compact structures known as strobili, or cones, which you can clearly observe in examples like Phylogenetic and Equisetum. Within these sporangia, specialized cells called spore mother cells undergo meiosis, reduction division, to produce haploid spores, N. These spores are the key to the next stage of the life cycle. Upon germination, these spores give rise to an inconspicuous, small but multicellular, free-living, and mostly photosynthetic thalloid gamophyte. This gamophyte is specifically called a prothallus. Now, this prothallus has very specific requirements for growth. It needs cool, damp, and shady places to thrive. Furthermore, water is absolutely essential for fertilization, as the male gametes are motile and need water to swim to the female gamete. Because of this specific and restricted requirement for their gamophyte to grow, and the continued dependence on water for fertilization, the spread of living pteridophytes is still somewhat limited, restricting them to relatively narrow geographical regions compared to, say, flowering plants. The prothallus, being the gametophyte, bears both male and female sex organs. The male sex organs are called antheridia, and the female sex organs are called archegonia. Water is required for the transfer of the antherozoids, the motile male gametes released from the antheridia, to the mouth of the archegonium. Once a male gamete fuses with the egg present in the archegonium, a zygote, 2N, is formed. The zygote then develops into a multicellular, well-differentiated sporophyte, 2N, which, as we discussed, is the dominant phase of the pteridophytes. Now, let's address the critical distinction of homosporous versus heterosporous, which is fundamental for understanding the evolution of the seed habit, a concept frequently tested in NEET. The majority of pteridophytes are homosporous, meaning they produce only one kind of spore, and all these spores are of similar kinds in terms of size and potential. They germinate to produce typically bisexual gamophytes. However, certain genera, such as Phylogenetic and Salvinia, these are key examples to remember, are heterosporous. This means they produce two distinct kinds of spores. One, macro, large spores, also called megaspores. Two, micro, small spores. The significance of heterospory is immense for understanding plant evolution. The megaspores germinate to give rise to female gatophytes. The microspores germinate to give rise to male gatophytes. Crucially, in heterosporous pteridophytes, the female gamophytes are retained on the parent sporophytes for variable periods. This retention is a pivotal evolutionary step. They don't just fall off and develop independently on the soil immediately. Why is this so important? Because the development of the zygotes into young embryos takes place within these retained female gatophytes. This event, the development of the embryo within the parent sporophyte's protected female gamophyte, is considered a direct precursor to the seed habit. It's an important evolutionary step because it provides protection and nourishment to the developing embryo, similar in principle to how a seed offers protection to a plant embryo. So, while pteridophytes don't produce true seeds, their heterosporous nature with retention of the female gamophyte is a clear evolutionary stepping stone toward the seed-bearing plants we'll discuss next. The Gymnosperms and Angiosperms. Pteridophytes are further classified into 4 classes, mainly for comprehensive understanding. Celopsida, examples like Celotum, like Copsida, examples like Phylogenella, Lycopodium. Synopsida, example like Equisetum, which is sometimes called scouring rush due to silica in its stems. And Pteropsida, the true ferns, examples like Dryopteris, Pteris, Adiantum. Remember, these examples for each class is helpful.
Okay, the concept of heterospora is frequently tested, and your explanation of it as a precursor to the seed habit is vital. Could you maybe elaborate just a bit more on why it's considered this precursor and perhaps its broader evolutionary significance for students preparing for competitive exams? It's a nuanced point that often needs, well, deeper clarity.
Yes, certainly. This is indeed a nuanced but high-yield concept for competitive exams. So let's elaborate on the significance of heterospory as a precursor to the seed habit. To reiterate, in heterosporous pteridophytes like Phylogenella and Salvinia, you have two spore types, large megaspores and small microspores. The key evolutionary innovation here lies in two critical aspects that directly relate to the seed habit. 1. Retention of the female gamophyte. Unlike Angiosperms pteridophytes, where the gamophytes develop independently on the soil, exposed to environmental hazards like drying out, in heterosporous forms, the megaspore, which gives rise to the female gamophyte, is largely retained within the megasporangium on the parent sporophyte. This means the female gamophyte, with its egg cell, develops and matures while still attached to and being nourished by the sporophyte. This provides a crucial, protected, nutrient-rich environment for the female gamophyte, which is a significant departure from the fully free-living gamophytes of Bryophytes and Angiosperms pteridophytes. Think of it as keeping the nursery attached to the parent plant. 2. Development of zygote into embryo in situ. Following fertilization, which still requires water for the motile male gametes to reach the egg, the zygote begins to develop into a young embryo within this retained female gamophyte, which itself is still enclosed within the megasporangium on the parent sporophyte. The embryo starts growing right there on the parent plant. Now, why is this a precursor to the seed habit? Let's connect the dots. Protection. The developing embryo is now shielded by the maternal sporophyte tissue. This offers a distinct advantage over free-living gamophytes, where the young embryo would be much more exposed to environmental hazards like desiccation or predation. This internal protection closely mimics the protection offered by a seed coat in true seed plants. Nutrient supply. The retained female gamophyte provides direct nourishment to the developing embryo, drawing resources from the parent sporophyte. This is a critical step toward the efficient provisioning seen in seeds, where abundant food reserves, like endosperm or cotyledons, are stored for the embryonic plant to use during germination. Reduced dependence on external water for early embryo development. While fertilization still needs a film of water, the subsequent development of the embryo within the protected maternal tissue significantly reduces the immediate dependence of the vulnerable young sporophyte on external water conditions for its survival and initial establishment. The dispersal unit effectively becomes something more robust than just a naked spore, offering a higher chance of survival. The broader evolutionary significance is profound. This move towards internal embryo development and retention represents a major step in the plant kingdom's full adaptation to terrestrial life. It sets the stage for the complete elimination of the need for external water for fertilization, achieved by pollen tubes and seed plants, and embryo development. It's an intermediate step showing how plants gradually evolved more sophisticated ways to protect their embryos and ensure reproductive success in increasingly drier environments, ultimately leading to the highly successful seed plants. Without heterospory and the subsequent retention of the female gamophyte, the evolution of the seed, as we know it, would likely not have been possible. It's a vital link.
That makes the link to the seed habit so much clearer. It's truly fascinating to see these evolutionary stepping stones laid out like that. Okay, now let's move on to gymnosperms, often simply known as the naked seed plants. What exactly does that term imply, and what are their general characteristics, including maybe their remarkable adaptations to harsh environments?
All right, let's transition to gymnosperms, a truly significant group that marks a major stride towards complete terrestrial independence. As you rightly put it, they are the naked seed plants. The name itself gives away their defining characteristic. Gymnos is Greek for naked, and sperma means seeds. So, in gymnosperms, the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. Consequently, the seeds that develop post-fertilization are also not covered by a fruit wall. They are quite literally naked seeds. This is the primary distinction from angiosperms or flowering plants, as we'll discuss next, and is a key point for your exams. Naked seeds equal gymnosperms. Gymnosperms include medium-sized trees, tall trees, and shrubs. A remarkable example that showcases their grandeur is the giant redwood tree, sequoia, which is one of the tallest tree species on Earth, a true natural wonder found on the west coast of North America. You can find them in diverse landscapes, from temperate forests to cold alpine regions. Let's look at their general characteristics, starting with their root systems. They generally possess a prominent taproot system, a main central root going deep. In some genera, like Pinus, pines, their roots form a beneficial fungal association called mycorrhiza. Here, fungi live symbiotically with the roots, helping the plant absorb water and essential nutrients, particularly phosphorus, more efficiently from the soil, especially in nutrient-poor conditions. In others, like cycas, a cycad often looks a bit like a palm tree, you find specialized roots called coralloid roots. These roots grow near the soil surface and are unique because they are associated with nitrogen-fixing cyanobacteria, remember those, which form a symbiotic relationship with the plant, helping it acquire essential nitrogen from the atmosphere. Their stems can vary. They may be unbranched, giving them a palm-like appearance, as seen in cycas, or they can be extensively branched, which is characteristic of familiar examples like pinus, pine trees, and cedar trees. The leaves may be simple or compound. In cycas, for instance, the large, pinnate, feather-like leaves are quite prominent and persist on the tree for several years, giving them a distinctive appearance. A very important adaptive feature, especially relevant for understanding their ecological success and for competitive exams, is how incredibly well-adapted gymnosperms are to withstand extremes of temperature, humidity, and wind. This allows them to thrive in diverse and often harsh environments. In conifers, which are a major group of gymnosperms, like pines, firs, spruces, the leaves are typically needle-like or scale-like. This needle shape significantly reduces the surface area exposed to the environment, thereby minimizing water loss through transpiration, which is crucial in dry or cold conditions. Furthermore, their leaves possess a thick cuticle, a waxy protective layer on the surface, and sunken stomata. Stomata, the pores for gas exchange, are recessed into pits in the leaf surface. Both of these are crucial adaptations to further reduce water loss and protect against extreme conditions, enabling their survival in drier, colder, or windy habitats. These adaptations are key to their widespread distribution, especially in northern latitudes and mountainous regions.
That's a great overview of their defining traits and remarkable environmental adaptations. Very clear. Now, let's get into their reproduction. You mentioned they are heterosperous, just like some advanced pteridophytes. Can you detail their reproductive process, from spore production to the development of the naked seed? And critically, how does their game-to-fight independence compare to keratophytes? This comparison seems like a really key distinguishing point for exams.
You're absolutely right. Understanding their reproductive strategy, particularly the comparison with pteridophytes regarding game-to-fight independence, is absolutely critical for conceptual clarity and exam success, as it represents that significant evolutionary leap we talked about. Gymnosperms are, in fact, heterosperous, meaning they produce two distinct kinds of haploid spores: microspores, which will form male gamophytes, and megaspores, which will form female gamophytes. This is a shared trait with advanced pteridophytes like Phylogenella, but the subsequent development and ultimate fate of these spores and the gamophytes differ profoundly. Let's trace their reproductive process step by step. The two kinds of spores are produced within specialized structures called sporangia. These sporangia are borne on specialized leaf-like structures called sporophylls. These sporophylls are typically arranged spirally along an axis to form compact structures known as strobilia or cones. These are the reproductive structures you typically see on pines and cycads. We have two types of strobilia or cones. One, male strobili or microsporangiate strobili. These cones bear microsporophylls and microsporangia. 1. Male strobili, or microsporangiate strobili. Within the microsporangia, the microspores are produced by meiosis. Here's a crucial point. These microspores develop into a highly reduced male gabophytic generation, which is confined to only a limited number of cells. This extremely reduced male gametophyte is what we call a pollen grain. The entire development of these pollen grains takes place within the microsporangia, which are retained on the parent sporophyte. This offers significant protection during development. 2. Female strobili, or macrosporangiate strobili. These cones bear megasporophylls with ovules or megasporangia. 2. Female strobili, or macrosporangiate strobili. In terms of cone placement on the plant, male and female cones or strobili may be borne on the same tree, which is the case for Pinus, making Pinus minucius both sexes on one plant. However, in Cycas, male cones and megasporophylls are borne on different trees, making Cycas dioecious separate male and female plants. This distinction between monoecious Pinus and dioecious Cycas is important for specific examples. Now let's look at megaspore and female gametophyte development in more detail. A megaspore mother cell differentiates from one of the cells of the nucellus, which is a mass of nutritive tissue within the ovule. The nucellus is protected by one or more layers called envelopes or integuments, and this composite structure, integuments plus nucellus plus megaspore mother cell megaspore, forms the ovule. These ovules are borne on the megasporophylls, which may be clustered to form the female cones. The megaspore mother cell, diploid, undergoes meiosis to form 4 haploid megaspores. Typically, only one of these megaspores, which remains enclosed within the megasporangium, nucellus part of the ovule, develops into a multicellular female gamophyte. This female gamophyte, unlike in pteridophytes, is never free-living. It develops entirely within the ovule and bears 2 or more archegonia, which are the female sex organs containing the egg cells. Importantly, this multicellular female gamophyte is also retained entirely within the megasporangium, within the ovule, on the parent sporophyte. Here's the critical distinction regarding gamophyte independence compared to previous groups, the point you asked about. Unlike bryophytes and pteridophytes and gymnosperms, the male and female gamophytes do not have an independent free-living existence. They are entirely contained, protected, and dependent on the sporophyte. The pollen grain, male gamophyte, and the female gamophyte with its archegonia remain within the sporangia or ovules, which are themselves retained on the sporophytes. This represents a monumental evolutionary advancement as it completely liberates them from the reliance on external water for gamophyte survival and development, which was still a limiting factor even for heterosporous pteridophytes. Finally, the fertilization process. The pollen grain is released from the microsporangium. It is then carried, usually by air currents, wind pollination, or anemophily, a crucial adaptation for land life, and comes into contact with the opening of the ovules, the micropyle, borne on the megasporophylls. A pollen tube, which grows out from the pollen grain and carries the male gametes. Sperm cells, which are typically non-mobile in higher gymnosperms like conifers, though cycads and ginkgo have flagellated sperm, then grows through the nucellus towards the archegonia within the ovules. It discharges its contents, including the male gametes, near the mouth of the archegonia, allowing fertilization to occur. Following fertilization, the zygote develops into a multicellular embryo, and the entire ovule structure develops into a seed. And as their name implies, these seeds are not covered by a fruit wall. They're naked, often borne on the surface of the megasporophyll scales, ready for dispersal by wind or animals. This entire process highlights the extreme reduction of the gametophyte phase and the increasing dominance and independence of the sporophyte, culminating in the production of naked seeds. These seeds are highly resistant dispersal units, allowing gymnosperms to effectively colonize drier terrestrial environments.
Right, the naked seed concept is definitely the hallmark of gymnosperms. Can you draw a clear, sharp comparison between the reproductive strategies of gymnosperms and pteridophytes, particularly regarding that gametophyte independence? Because that seems like a key distinguishing point that probably appears quite often in competitive exams.
Yes, absolutely. This comparison is vital for understanding plant evolution and is indeed a frequent point of examination. So let's clarify it with precision. The most significant distinction in reproductive strategies between pteridophytes and gymnosperms lies in the independence and retention of their gamophytes and the subsequent shift in their primary dispersal units from spores to seeds. Let's break it down point by point for comparison. Pteridophytes, especially the heterosporous ones, as they're the closest evolutionary link. 1. Gamophyte development and independence. While heterosporous pteridophytes show retention of the female gamophyte on the sporophyte, the male gamophyte, developing from the microspore, is typically shed and develops briefly. Even the retained female gamophyte represents only a partial step towards full dependence. The male gametes, antherozoids, are still motile and require an external film of water to swim to the archegonia for fertilization. This reliance on water for fertilization is a major constraint. 2. Embryo development. The development of the zygote into a young embryo occurs within the retained female gamophyte, which is this precursor step. However, the overall process is still somewhat vulnerable to external conditions. 3. Dispersal unit. The primary dispersal unit in all pteridophytes, whether homosporous or heterosporous, is the spore. The spore travels, germinates in a suitable moist environment, and then develops into the gametophyte, which leads to fertilization and the new sporophyte. There isn't a true seed containing a preformed embryo and food reserves that disperses. Gymnosperms. 1. Gametophyte development and independence. Here, both the male gamophyte, the pollen grain, and the female gamophyte are highly reduced and never have an independent, free-living existence. They are completely contained, protected, and nutritionally dependent within the sporangia, microsporangia for pollen, megasporangiovules for female gamophyte, which are themselves retained on the sporophyte. There's no free-living stage outside the parent sporophyte. 2. Fertilization. This is the game changer for terrestrial adaptation. The male gametes are delivered directly to the vicinity of the female gatophyte via a pollen tube, which grows from the pollen grain towards the ovule after pollination, typically by wind. This process completely eliminates the need for external water for fertilization. This is a massive evolutionary advantage for colonizing dry land. 3. Dispersal unit. The ultimate product of reproduction and the primary dispersal unit is the naked seed. This seed is a much more complex and robust structure than a spore. It contains a protected embryo, the unsporophyte, along with stored food reserves, all encased by a protective seed coat derived from the ovules and teguments. This allows for survival in much harsher, drier conditions, dormancy, and dispersal over longer distances. In summary, the key distinguishing point for your exams is this. Pteridophytes show an incipient or precursor seed habit with retained female gamophytes. But crucially, their motile male gametes still require external water for transfer, and spores are the dispersal unit. Gymnosperms, on the other hand, have completely lost the free-living gametophyte stage, have evolved wind-mediated pollination and internal fertilization via a pollen tube, eliminating the need for water for fertilization, and have developed the naked seed, containing embryo, food, and protective coat, as the primary, highly effective dispersal unit, truly liberating them from aquatic dependency for reproduction and allowing them to dominate vast terrestrial environments. This is the monumental evolutionary leap that defines the success of seed plants.
That distinction is incredibly sharp and crucial for exam success. Yes, that really highlights the evolutionary progress very clearly. Now, for the grand finale of our plant journey today, angiosperms, the flowering plants, these are truly the pinnacle of plant evolution, aren't they? They dominate so much of our terrestrial world. What is their defining characteristic that sets them apart from all other plant groups we've discussed, making them so incredibly successful?
Indeed, angiosperms, the flowering plants, represent the most diverse and evolutionarily successful group in the plant kingdom, dominating nearly every terrestrial habitat on Earth. They are the plants you interact with most frequently: grasses, herbs, shrubs, most trees you see daily. Their single defining characteristic, the feature that absolutely sets them apart from all other plant groups we've discussed, including gymnosperms, is this. Unlike gymnosperms, where the ovules are naked, in angiosperms, the pollen grains and ovules develop in specialized reproductive structures called flowers. And crucially, after fertilization, the seeds are enclosed within fruits. This enclosure of the seed within a fruit, which develops from the ovary part of the flower, is the undeniable hallmark of angiosperms. It provides an extra layer of protection for the developing seeds and often plays a crucial role in seed dispersal, whether by attracting animals to eat the fruit and disperse the seeds, or by modifications for wind or water dispersal. The evolution of the flower itself was a revolutionary step. It allowed for highly efficient and diverse pollination mechanisms, often involving coevolution with animals like insects, birds, and even bats. This targeted pollination is generally much more efficient than the wind pollination typical of most gymnosperms. This coevolution with pollinators contributed significantly to their rapid diversification and unparalleled ecological success across varied environments. Angiosperms are an exceptionally large group, occurring in an incredibly wide range of habitats, from scorching deserts to submerged aquatic environments, from freezing polar regions to lush tropical rainforests. They've conquered almost everywhere. Their size range is also extraordinary, from the smallest known flowering plant, Wolffia, which is just a tiny speck a few millimeters across, floating on water, to the towering trees of eucalyptus in Australia, which can reach heights of over 100 meters, rivaling even the giant redwoods. Their economic importance to humans is simply unparalleled. They provide us with the vast majority of our food. Think of staple grains like wheat, rice, and maize, countless fruits, vegetables, nuts, and pulses. They are also primary sources of fodder for livestock, fuel, wood, biofuels, medicines, a huge array of plant-derived compounds critical for pharmaceuticals, and countless other commercially important products like timber, fibers like cotton, jute, linen, oils, vegetable oils, beverages, coffee, tea, spices, and of course ornamental plants that beautify our surroundings. Our civilization heavily relies on them. Angiosperms are broadly classified into two major classes, which we will study in detail later, particularly regarding their anatomy and morphology. 1. Dicotyledons, or dicots, which typically have two cotyledons, embryonic leaves in their seeds, leaves with net-like reticulate venation, and flower parts usually arranged in multiples of 4 or 5. Examples include roses, sunflowers, mango trees, beans. 2. Monocotyledons, or monocots, characterized by a single cotyledon in the seed, leaves with parallel venation, and flower parts usually in multiples of 3. Examples include grasses, lilies, orchids, palm trees, bananas. This innovation of having their reproductive structures condensed into flowers leading to double fertilization, another unique feature you'll learn about, and ultimately resulting in seeds enclosed within protective fruits, coupled with their sophisticated pollination and dispersal strategies, has allowed angiosperms to colonize and dominate nearly every corner of the Earth, making them unquestionably the most successful plant group today.
So, just to crystallize this for our students, what is the single most definitive characteristic of angiosperms that truly sets them apart from all other plant groups we've discussed? The one thing that makes them angiosperms and has contributed so much to their success.
Yes, for your exams, the single most defining characteristic of angiosperms that you absolutely must remember is that their ovules are enclosed within a specialized structure called the ovary, which is part of the flower, and this ovary subsequently develops into a fruit after fertilization, thereby ensuring that their seeds are enclosed within fruits. This is the core distinguishing feature. Gymnosperms have naked seeds. Angiosperms have seeds enclosed in fruits. This innovation of flowers and fruits is the primary reason for their immense ecological success and diversity compared to all other plant groups. That's the key takeaway.
That's a perfect encapsulation. Excellent. We've covered an incredible amount of ground today, tracing the evolution and diversity of the plant kingdom, from algae right up to the flowering plants. Now, to bring it all together for our students preparing for exams, can you provide a concise summary of each group's defining features and perhaps highlight the key evolutionary trends they should keep in mind for conceptual clarity? Sort of a quick revision checklist.
Certainly. Let's recap the defining features of each plant group, then trace the significant evolutionary trends, which are crucial for conceptual understanding and for confidently answering analytical questions in your exams. This is a good way to tie it all together, starting from the most primitive to the most advanced in our discussion today, algae. Remember them as simple, thalloid, autotrophic organisms, largely aquatic. Key points for reproduction are their variations in sexual fusion isogamy, anisogamy, and oogotemy. Their immense economic uses as primary producers, half the Earth's CO2 fixation. Food sources like Porphyra, chlorella, and sources of hydrocolloids, algin from brown, carrageenan, agar from red, are high-yield topics. Make sure you can differentiate between green, brown, and red algae based on their characteristic pigments. Think of ETS-GLO-AT plus fucoxanthin-GL-AT plus phycoerythrin, specific stored food, starch, laminar and mannitol, Floridian starch, and flocculation patterns of their spores and gametes, flatulated in green, brown, non-motile, and red. Bryophytes. These are the amphibians of the plant kingdom because of their strict dependence on water for sexual reproduction and torso transfer. Their plant body is more differentiated than algae, being thallus-like or erect, attached by rhizoids, not true roots. The gametophyte is the dominant free-living phase, and it bears the dependent sporophyte. Don't forget their ecological role in succession and preventing soil erosion, and the economic importance of sphagnum moss, peat packing material. Be able to distinguish between liverworts, dorsoventral thallus, often with gemma cups for asexual reproduction, and mosses, distinct protonema and leafy stages in gabophyte, more elaborate sporophyte with spore dispersal mechanism. Pteridophytes, a major evolutionary leap. These are the first terrestrial plants to possess vascular tissues, xylem and phloem. Here, the sporophyte is the dominant independent phase, fully differentiated into true roots, stems, and leaves. Understanding the concept of homospery, most, versus heterospery, Selaginella salvinia, is crucial. Remember that heterospery is considered the precursor to the seed habit due to the retention of the female gamophyte and embryo development in situ on the parent sporophyte, although they still need water for fertilization and disperse via spores. Gymnosperms. These are the naked seed plants. Their defining characteristic is that their ovules are not enclosed by any ovary wall, and their seeds remain exposed post-fertilization. They are heterosperous, and the critical distinction is that their male and female gamophytes do not have an independent, free-living existence. They are entirely contained and retained within the sporangiovules on the sporophyte. They evolved wind pollination and pollen tubes, eliminating water need for fertilization. They exhibit excellent adaptations to drier environments. Needle leaves, thick cuticles, sunken stomata, angiosperms. The most evolved and diverse group, the flowering plants. Their defining feature is that pollen grains and ovules develop in specialized structures called flowers, and their seeds are completely enclosed within fruits, developed from the ovary. They exhibit double fertilization, unique feature, an exceptionally wide range of habitats and sizes, and are of immense economic importance. Now let's connect this to the key evolutionary trends to remember for your board and competitive exams, looking at the big picture. 1. Body differentiation. A clear progression from simple, undifferentiated thalloid bodies, algae, and bryophytes, to the development of true roots, stems, and leaves with well-differentiated vascular tissues, pteridophytes onwards, increasing structural complexity. 2. Reproductive independence from water, a gradual reduction in dependence on external water for sexual reproduction. Algae and bryophytes are highly water-dependent for gamete transfer. Pteridophytes still require water for fertilization. Gymnosperms achieve liberation from water for fertilization via wind pollination and pollen tube growth. Angiosperms further perfect this with specialized flowers and diverse pollination strategies, often involving animals. 3. Gametophyte-sporophyte dominance. A clear shift from a dominant, free-living gatophyte in bryophytes to a dominant, independent sporophyte in pteridophytes, gymnosperms, and angiosperms, with the gatophyte becoming progressively reduced, short-lived, and eventually entirely dependent on the sporophyte in seed plants. 4. Seed evolution. The progression from spores as the main dispersal unit, algae, bryophytes, pteridophytes, through the precursor to the seed habit in heterosperous pteridophytes, to naked seeds in gymnosperms, and finally to enclosed seeds within fruits in angiosperms. This signifies increasing protection, nourishment, and dispersal efficiency for the developing embryo, leading to greater reproductive success in diverse terrestrial environments.
It's a powerful summary, perfectly tying together the individual groups with the overarching evolutionary narrative. Fantastic for revision. Now, shifting to practical exam advice, what should our students specifically focus on for their upcoming board exams and NEET based on this chapter? Any tips to maximize their scores?
All right, for your upcoming exams, whether it's board exams or NEET, this chapter is absolutely fundamental and honestly a high-scoring area if you master it properly. I urge you to revise this chapter thoroughly, paying very close attention to specific details. Firstly, focus intensely on the distinctive characteristics of each plant group. This includes their typical habitat, the specific nature of their plant body, thallus, true root stem leaves, their characteristic pigments, the form of stored food, cell wall composition, and particularly their unique reproductive methods and structures. For instance, know the details of isogamy, anisogamy, oogamy in algae, the protonema stage in mosses, the prothallus in ferns, cones in gymnosperms, flowers and fruits in angiosperms. These are common sources of multiple choice questions. Secondly, always try to grasp the evolutionary significance of each major group. Why are bryophytes amphibians? Why are pteridophytes the first vascular plants? Why is heterospery important? Why are gymnosperms called naked seed plants? How did angiosperms become dominant? Understanding these underlying concepts is crucial for answering analytical and reasoning-based questions, which are increasingly common. Thirdly, pay special attention to the examples given for each group and subgroup in your textbook. These are frequently asked directly in MCQs and NEET and often appear in short answer questions or give example-type questions in board exams. For example, be able to recall specific examples of green, brown, and red algae, Chlamydomonas, Laminaria, Gelidium, or examples of heterosperous pteridophytes, Selaginella, Salvinia, or monoecious, dioecious gymnosperms, Pinus, Cycas. Create flashcards or lists for examples if that helps you. Finally, and this is crucial for exam success, practice past year questions based on this lesson. Actively seek out questions from previous NEET papers and board exams. Specifically, target questions that ask for differentiation points between groups. For example, practice distinguishing between red algae and brown algae based on multiple criteria. Compare liverworts and mosses, or explain the differences between homosperous and heterosperous pteridophytes, or gymnosperms and angiosperms. Drawing comparative tables yourself can be extremely beneficial for quick revision and solidifying these differences. This consistent practice will build your understanding, help you identify common question patterns and tricky areas, and definitely improve your speed and accuracy during the actual exam. Remember, consistent effort and targeted revision are absolutely key to excelling in biology.
Thank you so much for joining us for this really in-depth deep dive into the plant kingdom. We truly hope this structured lesson has provided you, our listeners, with immense clarity and confidence for your biology studies and, of course, for your upcoming exams.
Absolutely. It was my pleasure. Keep revising these concepts diligently. Remember, consistency in revisiting topics is key to mastering biology, especially for demanding competitive exams like NEET. And please feel free to reach out if you have any doubts about this chapter as you continue your preparation. Study hard and all the very best for your preparation and exams.